welcome to Planetterrian Daily, episode 118 for July 12, 2026. Let's get into today's research and health developments. Columbia researchers link serotonin signaling to faster progression of degenerative mitral valve disease in patients carrying a specific genetic variant. Columbia University scientists found that patients with degenerative mitral regurgitation who carry a particular genetic variant and take SSRI antidepressants show accelerated valve damage. The work identifies serotonin as a driver that worsens structural changes in the valve tissue. Researchers examined how medication choice could influence surgical timing in genetically susceptible individuals. The team is now examining whether altering serotonin pathways might slow progression in this subgroup. The study was reported on Science Daily. A research team in Sicily flew drones into the plume above an active volcano to sample gases near the crater rim using laser spectroscopy. The approach aims to detect changes in gas composition that precede eruptions. Early tests show the drones can maintain position in turbulent air while collecting data that ground instruments cannot reach. The work was reported on phys.org. Researchers intend the method to reach locations inaccessible to fixed monitoring stations. The project focuses on real-time plume chemistry rather than visual or seismic signals alone. Further flights are planned to refine gas ratio thresholds that might indicate impending activity. University of Göttingen trials found that cattle respond to virtual boundaries with the same avoidance behavior observed at conventional electric fences. The study measured turning angles and grazing distribution across multiple herds. The data suggest virtual systems can deliver comparable spatial control without physical infrastructure. Animals maintained consistent distance patterns whether the boundary was physical or projected. No additional training beyond initial exposure was required for the observed responses. The approach eliminates the need for wire installation or maintenance across large pastures. University of Potsdam researchers showed that standard extinction risk models perform differently depending on whether climate change removes a species' habitat or merely relocates it. Current assessment methods do not distinguish these scenarios, leading to systematic under- or overestimation of risk. The team calls for revised protocols that incorporate habitat dynamics. Models that treat all habitat change as outright loss produced higher risk estimates than those allowing for relocation. The distinction matters most for species whose ranges are projected to shift rather than shrink. Revised frameworks would require separate parameters for each type of change. From cardiac tissue changes driven by a signaling molecule, we move to another protein that moves toxic material between cells in the brain. Yale researchers identified a pair of neuron surface proteins that facilitate transmission of the toxic protein associated with Parkinson's disease. Blocking these proteins in mouse models sharply reduced spread of the aggregates and slowed disease progression. The finding supplies a new molecular target for therapies aimed at interrupting propagation rather than only clearing existing aggregates. Yale's identification of two neuron surface proteins that ferry toxic aggregates between cells offers a concrete handle on Parkinson's propagation. Blocking the proteins in mice cut spread and slowed motor decline, shifting attention from clearance of existing aggregates to interruption of cell-to-cell transfer. The next step is to determine whether the same surface proteins operate in human neurons and whether safe inhibitors can be developed. What remains unclear is how early in the disease this transmission begins and whether blocking it would still help after symptoms appear. Lund University surveys using trained detection dogs counted living and dead bumblebees along 60 Swedish road verges. Verges with dense flowers attracted foraging bees, yet mortality rose sharply where traffic volume was highest. The results quantify how roadside habitat can function as an ecological trap. Mortality counts were obtained through systematic searches aided by the detection dogs. High flower verges showed the largest gap between attraction and survival. The findings apply specifically to stretches carrying heavy daily vehicle loads. Low-dose exposure to sulfoxyl-4 changed activity of genes involved in reproduction in bumblebee tissues. The study links the next-generation insecticide to molecular changes that could affect colony viability over time. Because pollinators support roughly one-third of global food production, the findings add to concerns about sublethal pesticide effects. The work was reported on Science Daily. Gene expression shifts were measured in multiple tissue types after controlled exposure. The doses used were below levels that cause immediate mortality. The results point to longer-term effects on queen production and colony persistence. While protein surfaces control disease spread, other surfaces, those of gold atoms, actively reorganize to control chemistry. Researchers mapped how surface atoms on gold spontaneously form protective patterns that prevent oxygen from reaching the metal. The reorganization suppresses oxidation far more effectively than previously modeled. The mechanism explains the long-term brightness of gold objects and points to new ways to design gold catalysts for manufacturing and clean energy applications. Most people picture gold as a passive, chemically inert metal that simply refuses to react. In reality, atoms on its surface actively rearrange into ordered patterns that physically exclude oxygen, cutting oxidation rates by as much as 1 trillion times. Right now, any gold ring or watch you wear is protected by this dynamic atomic reorganization rather than by absolute inertness. The same surface behavior that keeps jewelry bright for generations also explains why gold remains an effective catalyst in industrial processes. One memorable detail is that the protective reorganization suppresses oxidation far beyond what static models had predicted. The practical takeaway is that future catalyst design can deliberately exploit surface atom mobility instead of treating gold as a fixed, unreactive platform. Engineers developed a robotic sensor that distinguishes gram-positive from gram-negative bacteria by surface mechanical properties alone. The system bypasses chemical staining and trained interpretation steps required in conventional gram tests. It is intended for rapid screening in food safety, environmental monitoring, and infection control. Classification occurs through direct physical contact without added reagents. The sensor provides results in a single automated pass. Developers aim to integrate the device into portable field units. From atomic scale self-organization in metals, we turn to engineered self-organization inside living cells. Postec engineers rebuilt the ribosome's protein synthesis machinery into an RNA-based platform that reads up to 6 intracellular signals and autonomously triggers a programmed response. The circuit lets cells act as simple decision-making units without external control. The work moves synthetic biology closer to autonomous cellular computers. The study was reported on phys.org. A genetic screen in Saccharomyces cerevisiae showed that no single PIF1 or RECQ family helicase is essential for tolerance to the G4 stabilizing compound pyridostatin. Sensitivity appeared only in double mutants spanning both families, and an additional helicase, SRS2, proved non-redundant for managing recombination. The results support a 2-layer model of G4 maintenance. The work was reported on bioRxiv. Single helicase deletions left cells able to grow in the presence of the compound. Only combined loss of representatives from both families produced clear sensitivity. SRS2 function remained necessary even when other helicases were intact. Reliable cellular decision-making depends on redundancy. Next, we look at how neural circuits achieve the same robustness. University of Hong Kong researchers working with Princeton and Columbia colleagues mapped sensory motor circuits in C. elegans that maintain function even when individual genes or connections are removed. The study demonstrates how backup pathways compensate for disruptions. The findings help explain why some neural defects produce little observable change in behavior. From biological redundancy that preserves function, we examine a material's failure mode where redundancy is absent. Researchers traced how soft lithium filaments fracture the rigid ceramic electrolyte, creating short-circuit pathways. The work supplies a mechanistic explanation for a long-standing failure mode. Solving the cracking problem could enable safer, higher-energy solid-state cells for vehicles and electronics. Experiments revealed cracks propagating through non-elastic simple fluids, challenging the assumption that only complex, elastic fluids can break. The observation forces a re-examination of the physical conditions required for fracture. The work may affect models of material failure in industrial and biological settings. The study was reported in Quanta magazine. Cracks formed and advanced under controlled shear without measurable elastic recovery. The fluids tested lacked the long-chain polymers previously thought necessary for fracture. The results indicate that fracture criteria must be broadened beyond elasticity. New geological mapping suggests Etna is sustained by magma rising through fractures created by tectonic plate motion, instead of fitting any of the three classic volcanic formation models. Confirmation would place Etna in a rare fourth category and imply that large volcanoes can arise through mechanisms previously linked only to small submarine eruptions. Mapping focused on deep structural features beneath the volcano. The proposed pathway does not rely on subduction-related melting. The findings reopen questions about how other large volcanic systems are supplied. From failure mechanisms at the atomic and materials level, we close with a broader look at how small molecular details scale to visible outcomes. Before we go, keep an eye on follow-up work from the Yale team on whether the same surface proteins operate in human neurons. That's Planetterrian Daily for today. If you enjoyed this, a rating or review on Apple Podcasts or Spotify really helps new listeners find the show. I'm Patrick in Vancouver. Thanks for listening, and I'll see you tomorrow. And if you'd rather watch than listen, find us on YouTube at NERA Network. Links in the show notes. And before you go, this show is part of the NERA Network, a family of daily podcasts on tech, science, and markets. Each one a short, sharp briefing you can finish on the commute. If today earned its place in your feed, here's your next listen. Unintended consequences, true stories of good intentions that backfired. Find every show free at neranetwork.com. Want the written briefing with source links? Each show keeps a summaries page on neranetwork.com, scannable, dated, and free. This episode used AI voice synthesis of my voice. Editorial selection and analysis are my own.